All right. Good morning, everybody. Today is Wednesday, May 25th, 2016. And this is Before the First Cup with Jules here on UCY.TV. Happy Wednesday, everyone. It is 75 degrees and sunny here in upstate New York. I don't know. I haven't been outside yet. I took the indoor route this morning, but it looks nice out the window. Where do we leave off yesterday? We titled the show DARPA, Pandemic, Shape-Shifting Vaccine, Synthetic Cures, and Biosensors. Yeah, it was a mouthful. We looked at a lot of different things over the last few days, but specifically that synthetic immune system that DARPA put out a proposal day for. And I saw the bidders went on April 28th and submitted their proposals and asked questions for building a synthetic human immune system for DARPA. So put on a seatbelt on that one. I'm trying to remember which story. I dug in deep after the show yesterday, as I always do. And I discovered that it not only was DARPA, but IARPA had something to do in there. And there was another DARPA division that I'd never heard of that is in there working on this stuff. And you know what? I don't think this is the time or the place. I'm still half asleep. But we'll look for it if I can remember. One warning, guys. Here we are spinning down the end of the school year. Summer vacation is coming to a start here very shortly. Homework winds down. And my responsibilities get greater. And it's going to become difficult this summer. So I'm not sure what I'm going to do or how I'm going to do it. I definitely don't think that I'm going to be recording at 5 o'clock in the morning every morning so I can get something on air if I can't go live. So I don't know. We might have to have a summer of repeats or something. But I just am pretty burned out. I do this 24/7 between my show and then the 30 other shows. And then all of, my garden, my plants, trying to take care of the yard. The spring is very difficult because I have all these hulking vines and things that all need to be tamed. And then everything else, all my issues with my family. So I just don't know, guys. I might need to chill out a little bit this summer. But I guess we'll see. But I'll tell you what, I am feeling it right now. And last few weeks have been hell on me, and I still am not getting good sleep, so whatever. All right, let's get back to this. I'm not sure what I want to talk about today. It's hard to get inspired some days, because every day is just another day on that hamster wheel. We talked about shape-shifting vaccines, synthetic DNA. We saw that there was a meeting at Harvard last week where 150 scientists were invited to Harvard to talk about creating a synthetic human genome. We know that they already have been editing human genomes in China and Sweden and UK. And they're talking about trying to find ways to edit permanently and evolve genes so they can get rid of disease that maybe were even genetic traits. Well, this was interesting. This came out of phys.org. Yesterday, U.S. biochemist wins award for rewriting DNA to mimic evolution. So we're giving doctors awards for rewriting DNA to mimic evolution. But see, evolution happens slow and purposefully to extend the traits that were beneficial to survival, and those traits work themselves out. You didn't have some designer standing there figuring out that maybe a pointy nose versus a rounded nose is going to make your cell line, your progeny survive longer. So now we give scientists awards for playing God, mimicking evolution. We already saw that in robots, which is a big caution. But here's the even bigger caution. See, evolution, we have a tiny little pinky toe. because we really don't need that pinky toe anymore, and eventually that little pinky toe is going to be gone. But you know what? Your grandchildren might now have a pinky toe with poison darts in it in the next generation or two, because some doctor who won an award decided that they were going to mimic evolution and provide extra special traits for the people who can pay for them. U.S. biochemical engineer Frances Arnold on Tuesday won $1 million Euro Technology Prize in Finland for her work on directed evolution, a method of rewriting DNA to improve medicines. Oh, okay. And develop green fuels. So it's all about improving medicines and developing green fuels. Of course, green fuels are where the synthetic biology reports from the DOE to Congress live. It's all being done in the name of climate change to make green. Frances Arnold received the 2016 Millennium Technology Prize in recognition of her discoveries that launched the field of directed evolution, which mimics natural evolution to create new and better proteins in the laboratory, says the Technology Academy of Finland. Arnold, 59, is a professor of chemical engineering at the California Institute of Technology, right in the lizard's den, and says her work is made possible to, quote, solve human problems, unquote. Yeah, wink. It's kind of like the Twilight Zone episode called To Serve Man. It ends up being a cookbook. Yeah, this is to solve human problems. Not our problems. The humans are the problem. Anyway, to solve human problems such as replacing toxic chemicals like fossil fuels. See, so this is under the Department of Energy, Biotechnology, Synthetic Biology Division. This makes me sick. Her method of creating new proteins with desired properties is being used to convert renewable resources like sugarcane into biofuels and to improve everyday products such as laundry and dishwashing detergents to enhance the performance in cold temperatures. You know, do you hear the way they put this little marketing spin? And oh, it's so wonderful. Look at what technology is going to do for you. But what they're not saying in any of this is, let's go back to Eric Drexler talking about desktop manufacturing. Let's think about Monsanto and how genes are genetically modified with viruses that have been sucked empty and little Trojan horse payloads have been put inside of them. So then the virus can go inside and look for any crack in the armor and invade the cell. And when I think about stuff like that, and I think about how they are using these technologies to invade biology, and not only invade biology, but then manipulate it in ways that not only do we not understand, but there's lots of sneaky things that they can do once they've invaded biology. They're also mimicking biology and copying biology. And that's where all the synthetic DNA comes in. And when they start mixing the synthetic DNA with the biological, like they were talking about in the New York Times in the story we read yesterday about the new shape-shifting vaccine to protect us from everything forever, any pandemics that come, universal vaccine against any bioterror agent. That is like the base levels of transhumanism happening right in front of our eyes, except they're just not calling it that. But they said in that story, in the New York Times story, that synthetic DNA that they made that shape-shifting synthetic immune system out of would literally go into the host organism's DNA and change it and merge with their DNA. Okay, that's called transhumanism. That is called being merged with the synthetic. And not that it's not already. We're going to look at some of that today because it's very possible that this really has been happening for 30 years since they've been slipping us the willy in our food since the 80s without saying anything. With mad science gone mad, genetically engineered food in the food supply. You gotta love when they've been feeding it to you for 30 years. And not, I'm sure in the 80s it wasn't huge amounts, but we found out about a few in the early 90s. I told you that's when all my weird allergies to food started, was back then. Every time I'd eat tomatoes, I'd get hives. And then we found out on the show. I didn't even know for 20 years later. I didn't know. And here we're doing research and we find out right around that same exact time, the Flavor Saver tomato came out, which was genetically modified on the market and nobody knew. And then they had to pull it. They probably had to pull it because it caused issues. I just know I got head to toe hives every time I ate tomatoes. I never had issues before. All right, anyway, back to this rewriting DNA to mimic evolution. So it sounds like such a good idea. Again, if we want to be able to have wings, we should have evolved with wings. Well, now you'll be able to pay for it. You know, this is where this is going to end up. As they're slicing and dicing and reacting to all this stuff and learning how to manipulate all of biology, control it, and make it do what they want it to do. This is enslavement. You know, these are the power brokers that own this technology, not you. Yeah, we have DIY CRISPR kits for Johnny to play with in the garage, but he's only doing yeast. E. coli for now. But eventually, who knows? Anyway, here we go. So, yes, we can make laundry and dishwashing detergents, enhance their performance at cold temperatures. So, all you housewives, this is learning how to do DNA evolution. It's going to improve your life. New and improve with new DNA evolved on the side of the box. They've changed sugarcane into biofuel, so no more sugarcane for your coffee. Maybe they should be using sugar beets. Why can't we use GMO sugar beets instead of putting that into people's foods? Hey, burn it in my car. I don't really care. The microbes convert plants to fuels. So the sugar that's in the plant is the feedstock. And instead of pumping oil out of the ground to make gasoline, we can now use sunlight stored in plants. Other practical applications, which, yes, what are we going to mine plants now? We're going to have plant farms where we could hold plants captive and steal their sunlight. I mean, maybe. I know that they've been messing with artificial photosynthesis. There were big algae farms a while ago where they'd have them set up on roofs. And they made tons of energy. And I'm trying to remember what they were pumping out. Because obviously it would be CO2, oxygen. But what other input-output? We had sun. I don't know. You know what? I'm going to make a note of that. The algae plants. Because those actually were pretty damn cool. And they produce tons of energy. And it's good for you, too. You know, any of those super greens, any of that stuff is great for you as long as it's not genetically modified or laden with poisons. All right, anyway, it says other practical applications of rewriting DNA to mimic evolution could be creams, fragrances, industrial solvents, or improving the drug industry's previously wasteful chemical processes to make medications such as Januvia which is used by millions of people to treat diabetes. So we're going to use this to pump out drugs. And this is pretty much the same process that they're talking about using now to circumvent any of the old ways that they've been doing genetic modification. And this is the way that they'll get around the labeling. And we have a few stories on this that I wanted to talk about today. Basically, what they're doing is using, and up until this point, they've been using like CRISPR technology to go in. We read about that. Edit E. coli to be able to zip and unzip. And then they program the cells, because you can do that now, just like a little computer program. You can program the cells so when there's an input, it produces an output. So, like she was saying, we give it some sugar and it produces some ethanol. Who knows? They can make it do all kinds of stuff. So instead of physically going in and modifying the base cell of the target, what they're doing is modifying bacteria that then they send in to go create chemical processes using something in the organism as a raw material to then pump out a new end resulting material. And hey, they didn't do any genetic modification. They just changed this little CRISPR E. coli gene and told it what they wanted it to do. And then they set him free. He went and did all the work then. Eventually, they're going to be able to do stuff like that in their body. And that's why I say be leery about any of these synthetic vaccines or synthetic GMO foods, because they could be putting stuff like that already into that we are then ingesting. And once it's inside of us, who knows what it would be able to do. I had a story here during the show that I don't think we talked about, but I put this in the notes of yesterday's show, but I wanted to touch on this. And this molecular robots. And I think this is pretty much what she is talking about. They can not only produce, but we can make them do things. This is out of the Weiss Institute, of course. This is the big pit of lizards who are making not only nanotech, but all these little DNA robot things and messing with proteins and building robo-bees. I believe Weiss Institute is tied to Harvard. Programming molecular robots. In the late 1990s, a small group of bioengineers set out to turn cells into tiny robots. Being bioengineers, they drew ideas from engineering and envisioned building a set of modular standard parts akin to sensors, power sources, microprocessors, and actuators that enable robots to sense and respond to their surroundings. And those early efforts spurred a wave of optimism about the incredible potential of synthetic biology. But even getting simple organisms to carry out the right tasks at the right time remained a formidable task, and synthetic biologists were still a long way from making living cells obey their commands. And that is because they only partially understand the workings of the cell's operating systems, their genes and their regulatory networks. And so a new contingent of bioengineers is pioneering a different approach. Instead of trying to program living cells, they're using the cell's information-carrying molecules, the DNA, the RNA, and the proteins, to build their own operating systems, their own sensors, and their own actuators. And their goal, well, to build tiny molecular robots. This past fall, the National Science Foundation kicked in $10 million taxpayer dollars from a National Science Foundation initiative called Expeditions in Computing to fund 11 scientists, including Weiss Institute core faculty member William Singh, PhD, Peng Ying, PhD, and former Weiss Institute postdoctorate fellow Sean Douglas, PhD, now an assistant professor of cellular molecular pharmacology at the University of California, San Francisco. Again, ding! Department of Energy, Pitt. Yes. So we're spending another $10 million of taxpayer dollars to fund the future of humanity's demise. It makes me so excited. We sat down with Singh, who was an associate professor at the Biological Chemistry and Molecular Pharmacology at Harvard Medical School, an associate professor of cancer biology at the Dana-Farber Cancer Institute, and Yin, who is an assistant professor of systems biology at Harvard Medical School, to learn about the ins and outs of this new branch of engineering, which could revolutionize fields such as information technology, ding, tissue engineering, ding, and manufacturing. Guys, this is... I mean, honestly, I don't even have words to express how disturbing this is to me, and it should be to you. We now, our cells that make up our body are going to become component parts in manufacturing. There will be no more factories. Get it? I just saw now not only is HP, Intel, and Microsoft all having huge layoffs, but now IBM announced them. Hardware, manufacturing, tangible items are starting to move by the wayside. Extra cash to spend on those things is as well. But they are moving to this on-demand, software as a service, everything in the cloud, 3D print, biosensing, real-time world that is going to be your fully medicated white room, brave new world. All right, let's look at a few of these questions. I don't know if I'm going to read this whole interview, but there was some interesting stuff in here. And this Weiss Institute, home of the Robo-Bees, is at it again. Yin, what is molecular programming? Yin says it means directing information-carrying polymers, i.e. DNA, RNA, and proteins, to demonstrate prescribed molecular behavior. Using these tools, we want to develop real-world applications. For example, we are using nanoscale DNA structures as a template to fabricate inorganic materials. So we're using DNA to build an inanimate object of some kind. We're also building programmable molecular machines made of DNA or RNA to help us study biology. I'm sure. But we have very fast computers these days. Why is it important to build programmable devices out of DNA, RNA, and proteins? Well, Seek says if molecular programs could only do an extremely slow job of what silicon computers can already do efficiently, they would only be a curiosity. But for crunching millions of numbers, they will never compete with these machines. I can think of at least two practical reasons for molecular programming. First, sometimes you need your computer to talk to cells, and you need to talk to molecules. You need your computer to diagnose a disease. You don't need to do billions of calculations per second, just maybe 10 in an hour, but you need your computer to go to small places in your body and to be biocompatible. Ding! Transhumanist future, everybody. The second reason is that you can create massively parallel armies of not very good computers. Yeah. And then you can genetically modify them. For example, a traditional strategy for drug discovery entails taking a molecule that works and then tinkering with it and making it work better. But a second strategy that has not worked well so far is to make billions of drugs and then screen them and build off of that. But instead of making a random library of new drugs, what if we had a way to build in logic? Create a library that is more streamlined and more of the things that you want. The critical thing for us is that we're not trying to use these molecules to do mere computing. We are trying to use them to perform programmable molecular tasks. For example, we can program how they physically interact with each other. As information carrying molecules can specify spatial and dynamic behavior. And one example is digital fabrication. We use a programmable DNA structure to specify the nanoscale 2D and 3D geometric shape. And then we can translate from those 2D and 3D shapes to other inorganic substrates. For example, we can translate DNA shapes to a gold particle and then later to graphene and then create a substrate for nanoelectronics. We're going from programmable biomolecules and then using them to fabricate inorganic devices with a digitally programmable shape. What? What did that just say? Oh my God. They are using biological DNA, RNA, and proteins to then create, through reactions, produce a substance that then they can use to fabricate some sort of inorganic nanoelectronic. I don't know. But see, the scary thing is this Weiss Institute is full of lots of globalists. And they're all part of the Skull and Bones group that are working very closely with DARPA, Department of Energy, synthetic biology, mini drones and robotics. This is where the origami manufacturing came from, to be able to make robo-bees by the bazillions. Yeah, that Weiss Institute. And now they're building molecular machines. Remember, we had out of MIT, Eric Drexler, the father of nanotechnology. And this is nanotechnology being realized here. Except it's even more than all the nanotech that I've learned about since the early, it's probably 89, maybe 1990 cover of Scientific American. I'll never forget that cover. I read that and Discover Magazine religiously every issue. But I learned about nanotechnology right about 1990. I've been paying attention ever since. And I never in my wildest dreams did I expect nanotech to include biological. Okay, it was always little mini robots. But I guess I never thought about what the robots are made of. So they're building little mini nano robots with biological components to make inorganic or organic. It's just frightening. They can make anything. This literally gives them the power to manipulate at the building blocks of life scale. It says you and your colleagues write in your grant application that with molecular programming, chemistry will become the new information technology of the 21st century. How is that? Yes, chemistry sure will. It says it's not going to be CPUs and smartphones or here or there, and points to a screen in his computer. But what if what you want is ubiquitous computing? Oh, we've talked about ubiquitous. That means everywhere and anywhere. We talked about wearable computing. We mentioned that new biosensor. It's going to be able to take your electrical and your chemical readings at any instant of the day and send it on to somebody outside of your body. Well, guess what? This isn't even wearables. This is ubiquitous. Everywhere. Trillions of sensors, he says, all over the body. Maybe with molecular computers, you could do it. Or you could disperse these sensors all over a lake or fill up the atmosphere. And collectively, they are doing something you cannot do with conventional silicon. This is the new frontier for computing. See, they've merged. I knew they merged. It is my industry merging with chemical and biological to become this transhumanist future. People, this isn't a joke. I mean, I know I laugh. I know I'm sarcastic over this because it's the way I deal with my worst nightmare coming true. Every morning I do my therapy by coming on and doing the show. Helps me get it off my chest so I don't walk around ready to explode with nobody to talk to about this because it bothers me so much. How are real world problems? What are real world problems that molecular programming could solve? Well, Yin says molecular programming could lend molecular precision to diagnostics or therapy. Of course, it's all about diagnosing. Life at its finest scale can now be visualized as dynamic, self-assembling molecular systems. And to interact with this molecular system, you want instruments on the same scale. You have digitally programmed instruments on the molecular scale. For example, there could be genetically encodable RNA machines to identify cancer cells and kill them inside of you, I'm sure. Or you could have a tiny fluorescent DNA probe that interacts with cellular components to produce super sharp images, so little cellular cameras. Digital fabrication is also extremely exciting. Using DNA structures as templates, you could specify in a digitally precise fashion the architecture and potentially the function of a nanoelectronic circuit or nanophotonic device. Now, photonics, it's scary because that's the way that they can add and remove memories. Remember, flashes of light. It says we're also collaborating with Pam Silver's group to assemble RNA bricks into an RNA scaffold to spatially organize enzymes involved in hydrogen production in E. coli. Ding! Using an earlier RNA scaffold, Pam's lab could increase hydrogen production by 50%. Pam Silver is a founding core faculty member of the Weiss Institute, so she's one of the lizards. And a professor of systems biology at Harvard Medical School. But, William, you've developed DNA origami. Ding, ding. Now, that was all done as part of the whole RoboBeast project, guys. Again, I followed that one from day one. You have developed DNA origami, and you use it to fold DNA strands into a bridge with handrails and other shapes and do all kinds of cool stuff. Is your molecular programming a continuation of your DNA origami work? Well, one connection is that the way people have done DNA computation so far is to have lots of free-floating strands of DNA in a solution, and they sometimes bump into each other, but the process is slow. So to catalyze the reaction, what if you sequester strands in a compartment or walking along the surface? For example, you can make a nanoscale box and then put a small number of strands into that box. And the effect of concentration could be up to 1 million times higher. And here's the last question, guys, and then I got a couple other things that we need to look at here. What do you envision is the future of molecular programming? Yin says, my mom was a computer engineer in the early days of punch cards. And now I can just type in Java and C, and I can program my computer. What we want to do is We want to do that for molecules that can carry then digital information. We want a user-friendly programming interface and an associated molecular compiler so that everyone, remember, democratic, everyone can do molecular programming. I don't see why, he says, in the future we couldn't imagine molecular apps and build the infrastructure to make them programmable, expressive, robust, and functional, and then many, many other users could really develop on their own apps. In 10 or 20 years, freshmen at Harvard could learn a molecular programming language, how molecules behave, and generate their own molecular apps that would be fascinating. Way worse than DIY CRISPR. These information-bearing molecules, he says, are merging with information technology and with molecular technology to enable human beings to realize our functional needs in the molecular world. This vision, I think, is an emerging revolution. You know what this sounds like to me? This sounds like to me like the beginnings of the replicator. We're going to be able to control, program, manipulate at the molecular and the nano level the building blocks of life, guys. And then we're going to democratize it and give it to all the freshmen. All the freshmen go out there and start slicing and dicing and creating freaks of nature. We had this doctor already winning $1 million prize for being able to fake out cells for evolution. To make creams and fragrances, detergents and laundry soap. They're working cold temperatures. Yeah, those are some pretty noble goals. What they're not telling you is that this is all going to be used to not only enslave you, but to get rid of us, to make better humans, to make better biology, to create psychotic visions of a living hell. I mean, this is really going to the pits of every worst nightmare that there's ever been. This is not going to be used for good, for benefit. These people might all have good intentions. But remember, there's military money that is being involved in all of this. Department of Energy, Department of Defense, Bayarda, or whatever their name is. A lot of this is being done in the name of keeping you safe. You know, we read about all the synthetic immune system programs yesterday, and we took it back at least a decade to that Blue Angel Project, making universal vaccines, enslaving tobacco plants to do our bidding, and to create these molecules, express these antibodies that then they could take and put inside virus-like Trojan horse particles and hijack you. Hijack biology by sneaking them past the immune system and having them invade your cells. Guys, this is nefarious. It's all being done in the name of, wow, look at how amazing, dangling carrots, new technology. People are just tra-la-lying along because it's all going to be done in the name of precision medicine and climate change and greening up the planet. Let's keep everybody from dying because, hey, we're headed towards a 6th grade extinction. But the joke is on us. We're not headed toward the 6th grade extinction because anything we did. We're headed toward the 6th grade extinction because of all the mad science gone mad that came long before the mad shit we're looking at now. The wanton pollution and dumping and not caring about any of it. Not believing in any balance or any type of other consciousness or ecosystem. We just said, fuck it. It's all for profit. Let's just use her up and dump her. Pick up the next one. And now they've moved on. Now that they've ruined the environment and the soil, the water, the air, they've moved on to our food. And now, hey, that's out of the bag too. Can't put that genie back in the bottle. Now we're on to cells. We did the genome project. We got humans. We got plants. We got animals. We got insects. And now we're making programming languages and programming at a molecular, cellular, and nano level. This bothered me. When we first read about those memristors, we were like, what? Well, this is even better. Performing cellular surgery with a laser-powered nanoblade. Yes, there are nano blades now. To study a certain aspect of cells, researchers needed the ability to take the innards out. So kind of like we were doing the viruses, we sucked them out. And then they needed to manipulate those innards that they sucked out of the cell and then put them back in. This is exactly what we've been talking about. It's how they do this magic mojo that they do to make the Trojan horse payload for these new genetically modified foods and for these new synthetic immune systems. Anyway, options for this kind of work are limited. But researchers reporting May 10th in Cell Metabolism, which is a research publication, described a nanoblade that can slice through a cell's membrane to insert mitochondria. Two researchers have previously used this technology to transfer other materials between cells, and they hope to commercialize the nanoblade for wider use in engineering. So, hey, you know what? This is going to make it so much easier now to suck the guts out of cells and replace them with whatever the hell we want. It's a new tool for cell engineering to truly engineer cells for health purposes and research. I think this is very unique, says Mike Titiel, a pathologist and bioengineer at the University of California at Los Angeles. We haven't run into anything so far up to a few microns in size that we cannot deliver. Titiel and Pingyu, also a bioengineer at UCLA, first conceived the idea of a nanoblade several years ago to transfer a nucleus from one cell to another. However, they soon delved into the intersection of stem cell biology and, energy metabolism, and that's where they started messing with the mitochondria, where the technology could be used to manipulate the cell's mitochondria. Studying the effects of mutations in the mitochondrial genome can cause debilitating or fatal diseases in humans is tricky for a number of reasons. This nanoblade apparatus consists of a microscope at a nano level, a laser at a nano level, and a titanium-coated micropipette to act as the blade, operated using what? Oh, you guessed it, a joystick controller. When a laser pulse strikes the titanium, the metal heats up and it vaporizes the surrounding water layers in the media, forming a bubble next to the cell. And within a microsecond, boom, the bubble expands, generating a local force that punctures the cell membrane and creates a passageway for us to invade it. That then we can pass cargo through, in this case mitochondria, and then the cell rapidly repairs the membrane defect. The team's aspirations go way beyond mitochondria, and they've already scaled up this nanoblade apparatus into an automated high throughput version. So there we go. Now it's going to be ready for primetime manufacturing. Large scale cellular surgeries. We wanted to make a platform that is easy for everybody to use. Oh my God. And allow researchers to devise anything that they can think of. A few microns or smaller that would be helpful to their research. Whether it's inserting antibodies or pathogens or synthetic materials or even something else we have not even yet imagined. Oh, it would be so cool, says TTL, to allow people to do something that they can't do right now. I think these people have some sort of mental defect if they think that this is good science. How can you think? I mean, all right, it's cool. You did it in the lab, but let's democratize it and give it to the whole world now. To come up with, I don't know, inserting synthetic materials or pathogens or who knows, anything, things you haven't thought of yet into cells. To completely destroy what Mother Nature created. Oh, but we're making it better. Shh, don't worry about it. We're going to engineer new humans. 21st century. It's what it's all about. And we're going to make you live forever. You're going to have your biosensors. There's going to be trillions of them in your body. We'll know every second. And don't worry, good old Billy Gates is working on synthetic biotics to be able to manipulate and control you through your gut bacteria. You know, and this is what worries me. So we have now, I saw yesterday Monsanto rejected the bid that Bayer made to purchase it. That was late last night. That doesn't mean it's over. It just means that they didn't think they were paying them enough. But yes, there's a potential merger between Bayer and Monsanto. And you know, Bayer was involved all the way back in the Nazi Germany making chemicals. So, yeah, that's a great company to be involved with Monsanto. And Monsanto's already been messing with genetically engineering plants and now moving on to this next generation of invading cells and RNAi stuff and putting it into food. We've also looked at the whole glyphosate issue, spraying that everywhere, and how badly it not only decimates your gut bacteria, but it completely takes your body out from all kinds of angles, whether it be gut or immune or memory or disease. So we're going to take a step forward. So another chemical company, and I don't know, maybe we'll look at the Bayer. I'm just looking at the time now. There's only like 8 minutes left, so I don't really want to get into the Bayer Monsanto story today. Maybe tomorrow as a part of what we're going to look at now. So I wanted to look at some of this new CRISPR and what they're doing to food and some of the things that really disturb me about this. Because in the past, we've already found out, and we've done 2 or 3 shows on edible vaccines, something that I never knew about. I heard about from one of the shows that April and Verstein had done in Truth or Talk. And I was like, what? Edible vaccines? No. And then started digging into it. Oh, yes. Remember, all this new stuff is about putting it in you. Okay? Making you part of the Internet of Things at a cellular level. Here, we'll start here. Banana vaccines. A conversation with Dr. Charles Artston. And this is out of 2005. So this is already 11 years ago. As Archimedes could attest, inspiration can strike anyone, and blah, blah, blah. When Charles Artston of Arizona State University. And by the way, that's where a lot of the synthetic biology is going on. And there's a lot of money from the feds going into Arizona State. We've read about quite a few of those already. Charles Arston of Arizona State visited Thailand in 1992. He was not expecting a moment of scientific eureka that would redirect his career. However, after observing a young Thai mother soothing her fussy infant with bits of banana, this plant molecular biologist was struck with an idea that is both startling and ingenious. What if, and this is thinking along the lines of Bill and Melinda Gates with malaria goats, what if, in addition to quieting her child, this mother could also be administering a life-saving vaccine in the banana? Well, from vision to reality, Armstrong's vision is well on its way to becoming reality, and this was in 2005. Thanks to a combination of dedication and applied biotechnology. As the president emeritus of the Boyce Thompson Institute for Plant Research, founding director of the Arizona Biomedical Institute, and member of the President's Council of Advisors on Science and Technology. So this guy's sitting up already in the White House advising. Back in 2005, it was Georgie Porgy Bush about Project BioShield. Don't worry about it if they're not going to take it. We'll just put it in their food. His breakthroughs lie in forging a link between plants, foreign DNA, and vaccines. Hey! So, I was talking about in 2006, me taking that FEMA class and them telling us about the universal flu vaccine, which was part of that Blue Angel program. And now we find out it's gone way beyond flu. It's become synthetic immune system. And they use plants to express these antibodies to make vaccines. And hey, here's the granddaddy of it all. This was in 2005. He started his research in 92. Vaccination stands as one of modern medicine's greatest success stories, and early experiments show that it could prevent disease merely by exposing a patient to a weakened or inactivated pathogen. And while his protocols violate today's clinical trial regulations, right, because clinical trials, remember, ethical issues, unless there's an emergency, and we now call out Project BioShield, or we pass the 21st Century CARES Act, where we don't need to have FDA approval. The dead and the dying can sign right up, and we'll do a case study if it works out well. But back then, they still had some ethics on the books. Dr. Jenner was able to prevent children from getting smallpox, even when he deliberately exposed them to it. What? Oh, God. He deliberately exposed children to smallpox, but hey, don't worry. He first inoculated them with pus from the cowpox. Today, most new vaccines contain specific proteins or a set of proteins from a pathogen of interest and not the pathogen itself. A protective immune response then can result from this more limited exposure, and though materially different from those developed by Jenner and Pasteur, modern vaccines, including Armstrong's, still build upon that same fundamental principle. If the immune system is trained, or wink, now made fully synthetic and shape-shifting, and to recognize a pathogen prior to infection, the disease can be prevented when the actual pathogen is encountered. Well, while Armstrong's edible vaccine is likely to win approval from children everywhere, there are actually significant medical advantages to this route of administration. And remember, when we dug into digestion and some of this stuff before, we discovered that there were two different ways that it was very easy for them to get nanotech and things of that nature into our bloodstream. One was intranasally, to cross that blood-brain barrier, and two was through the small intestine in the gut, so to be absorbed into the bloodstream. So edibly. Anyway, there are two significant medical advantages to the route of administration through edible vaccines. An oral vaccine incorporated into a plant bypasses the need for sterile syringes, costly refrigeration, and multiple injections. And furthermore, since many of the world's developing nations have the world's most deadliest diseases, cholera, rotavirus, E. coli infections, to name a few, they enter the body through the gastrointestinal tract. A vaccine that is ingestible may actually provide the best protection because it mimics the natural route of infection. The trick with an edible vaccine is convincing a plant to express the genes of a foreign organism. Unfortunately, Arnstein's work prepared him to face this challenge. As a biochemist, he focused his career on unraveling the means by which plants capture solar energy. And so what we've done for the last 10 years is to try to change the cellular machinery of a plant by adding new genes that can then make new proteins and coax those new proteins into folding to the desired shape so that it accumulates. And he took the knowledge about plant proteins and used that for something new, and that was for new vaccine development. They had already, in the 1990s, started investigating using genetically engineered yeast to produce proteins for injection into vaccines. And Arnstein's experience with green plants led him to consider other options. Would it be possible to use a higher plant instead of a lower plant? Can we make a potato or a tomato and turn it into a factory? And that's where we're going to have to stop for today, because we're going to have to look at the rest of this tomorrow, guys. All right, make sure you stick around because it's Wednesday. Lonnie Clark is live up next. And then we have Josh live at 6 p.m. Eastern. The boys from New York at 8. Liam Shuff, the brand new show at 9. And live at 11, Anti Media Radio with Nick and DB. All right, guys, have an awesome Wednesday. I will catch up with you tomorrow, everybody.